This is the presentation on cells, specifically Chapter 3 in Wholes Human Anatomy and Physiology. So you've probably been told this before, and I expect you studied this in general biology in high school at some point, but cells are the basic units of the body. The cell is the basic unit of structure and function. And cells are extremely small. We measure them in micrometers, shown by the mu sign, or you could just put a U and then an M. Cells that have developed specialized characteristics are said to be differentiated. So each of the cells in your body is different from each other in several ways, even though they may have exactly the same genetic material. Cells also vary in size and shape, and their structure and function are interrelated. And as we've said before in anatomy and physiology, as you know, function follows form. And that's true even at the cellular level. Okay, this graphic shows some different types of cells. As you see, they're very different in shape and also size. You look at the numbers there. This is kind of the basic cell you think of when someone says a cell, but could look like this. This is type of muscle cell, specifically a smooth muscle. And this is nervous tissue. This shows different types of epithelium. It can be flat in one layer. It can be multi-layers. And then some types of muscle tissue is actually branched, like in the case of skeletal muscle. But as you can see, there's no typical cell, but a composite cell shows the components found in most cells. So that's what we're going to study, and that's what you need to commit to your memory. So there's three major parts of a cell: the nucleus, that contains the genetic material. It directs cells' activities. When we draw it, we usually show it as a sphere in the middle of the cell. The cytoplasm, that's kind of the liquid stuff, but actually what we call the liquid stuff has a lot of organelles in it. It's really packed. They're suspended in liquid. The liquid itself really is called the cytosol. And then all of this is surrounded by a cell or plasma membrane. It's the outer boundary of the cell. It maintains the cell integrity, controls the entry and exit of substances, and we'll talk a lot more about that. Okay, and this is kind of the basic composite cell we usually show students when we talk about cells. Now, specifically, this is an animal type cell. And we're going to go through each of these types of organelles. But this is the image I would start working with if I were you in memorizing the parts. You're also going to have to know the functions of each of these organelles, which we will go through. So first of all, the cell or plasma membrane. It's the outer boundary of the cell, as we've already said. It maintains the integrity of the cell. It's very flexible and it's thin, but it is elastic. And it separates the intracellular fluid, as I said, the cytosol, from the extracellular fluid. Now, here's a term we're going to talk about quite a bit, which is selectively permeable. That means the membrane regulates what goes into it and out of it. Also, cell membranes are responsible for signal transduction. That permits the cell to receive and respond to messages. And the membrane consists mostly of lipids and proteins and a few carbohydrates. Okay, the lipids are actually phospholipids. That means they do have a phosphate part to them, and it is a bilayer. It has a water-soluble or hydrophilic head that forms a surface, and water-insoluble or hydrophobic tails that form the interior of the membrane. Okay, and these heads are on both sides of the membrane since it's a bilayer. The bilayer is permeable to lipid-soluble substances, since it does have lipids in it, but not to water-soluble substances. And something we talk about often in chemistry and biology is that like dissolves like. Okay, so lipid-type substances can dissolve or mingle with lipid-type substances, but lipid and water-soluble cannot intermingle. They do not mix. Cholesterol stabilizes the membrane. It helps keep it impermeable to water-soluble substances. Some lipids and proteins actually move around, and we call this a fluid mosaic of the cell membrane. It's very much like a bunch of balloons floating on top of the water in a way. Then you have membrane proteins with many functions. Some of them are pores, which allow things to move in and out, or channels also. Receptors, enzymes, cell contact and identification, and cell adhesion molecules. Now, we said there's some carbohydrates in there too, just a few, and they're for cell recognition and interaction. They're self-markers. So here's the lipid bilayer we're talking about. You have those polar heads of the phospholipid, and you have the nonpolar tails. Okay, the nonpolar tails are the hydrophobic, and the outer part is hydrophilic. But again, and hydrophilic means water-loving. But since the interior is hydrophobic, water cannot just simply move through the membrane. Okay, here's a more detailed image of the bilayer. Again, you see the heads on the outside, the tails on the inside. Here we have a cell identity marker. Okay, this shows carbohydrate chains. They may be for identification. Here's a receptor protein. Here's a channel protein. Here's what we call integral proteins. And every here and there you see a few cholesterol molecules. Remember, it said that it stabilizes. Okay, so as far as protein types, we have receptors that respond to extracellular signals. We have porous channels carriers that transport molecules and ions, and also they transduce signals. We have enzymes. Those are proteins. They catalyze chemical reactions, so they enable the reactions to occur. They speed them up. We have cell surface proteins that establish itself from non-self, so other parts of your body recognize itself. This is kind of very important for like white blood cells, so they don't attack your own body. Cellular adhesion molecules, they enable cells to stick to each other. Okay, let's look at a clinical application. Well, sometimes ion channels don't work exactly right, so a faulty ion channel can cause certain diseases. So mutations in the sodium channels, they can cause inability to feel pain or extreme pain conditions, the opposite. We have mutations in the potassium channels that disrupt the electrical activity of the heart. They disturb the heart rhythm, but they can also impair hearing. Abnormal chloride channels, these cause cystic fibrosis. So these individuals will have production of thick mucus, which causes difficulty breathing, a clogged pancreas, and salty sweat. Okay, cellular adhesion molecules, or HAMs. So CAMs guide the cells on the move. Selectins, they coat the white blood cells and anchor them to capillary walls by providing friction. Adhesion receptor proteins, they bind to the integrins to help white blood cells leave the capillary. The integrins direct white blood cells through the capillary walls toward the infection sites. And then there are other CAMs. Some guide embryonic cells towards the maternal cells to form the placenta. Some establish connections between the nerve cells. Okay, so this graphic shows some cellular adhesion molecules. So organelles do occupy the cytoplasm, but let's talk about the cytoplasm as a whole first. It consists of a network of membranes and organelles that are suspended in the cytosol. So cytoplasm is cytosol, which is the fluid portion, and the organelles. And the organelles are the solid structures with specific functions in the cell. The cytoplasm contains a cytoskeleton, which is a supporting framework of protein rods and tubules. Individuals have compared it to like lots of cross sections of spider webs in a way, but much, much smaller. When you look at cells in the microscope, you will not see the cytoskeleton. Usually you have to have something like electron microscope. The nucleus of the cell is considered to be an organelle. Okay, ribosomes. They're composed of protein and RNA. They can be free in the cytoplasm, or they're found attached to endoplasmic reticulum. Then we call it rough endoplasmic reticulum, or RER. The ribosomes provide structural support and enzyme activity to link amino acids in protein synthesis. The endoplasmic reticulum. These are membrane-bound sacs or canals and vesicles. It's a tubular transport system. The rough ER contains ribosomes, so it conducts protein synthesis. The smooth ER does not have ribosomes, but it conducts lipid synthesis. So this is a picture of ribosomes in endoplasmic reticulum. So this is our drawing of what it would look like three-dimensionally. As far as actually seeing in electron microscope, this is what it ends up looking like. Okay, vesicles. These are membranous sacs that kind of float around in the cytosol, and they store transport substances. Very important. The Golgi apparatus. These are sacs of flattened membranous sacs, usually outside of where the endoplasmic reticulum is. The Golgi apparatus refines, packages, and delivers proteins that were made on the rough endoplasmic reticulum. An example of organelle interaction, milk secretion. This is an example of interaction among the rough and smooth ER, and the Golgi apparatus, and transport vesicles. Here we have a three-dimensional image of the Golgi apparatus. Notice it's outside the rough endoplasmic reticulum. And here we have the electron micrograph. So this shows how these membranes, these organelles might interact. So the rough endoplasmic reticulum would produce milk protein. Then they send it to the Golgi apparatus. And sugars are synthesized in the smooth endoplasmic reticulum and the Golgi apparatus, and it can be attached to the proteins or secreted in the vesicles. But in any case, the proteins here in the Golgi apparatus will be modified. Then they're sent out by the vesicle, and the vesicle will meld with the cell membrane, and then that milk will be released. Okay, other organelles, the mitochondria. These are membrane-bound, fluid-filled sacs. They house chemical reactions that extract energy from nutrients. So this is where we say cellular respiration occurs, and that produces ATP, which is energy. So we call the mitochondria the powerhouse of the cell. Okay, lysosomes. These are small membranous sacs. They contain enzymes that digest proteins, carbohydrates, nucleic acids, bacteria, debris, and worn out cell parts. So they're kind of like the garbage disposals of the cell. And then we have peroxisomes. These are membranous sacs that are similar to lysosomes, but they contain enzymes that digest specifically lipids, alcohol, hydrogen peroxide. Here we have an image of mitochondria as an electron micrograph. On the right side, you see some of the lysosomes. Okay, other cytoplasmic structures. These things form the cytoskeleton. And the cytoskeleton holds the organelles into place quite often. Okay, these are made up of microfilaments, microtubules, and intermediate filaments. They are thread-like structures in the cytoplasm that create the cytoskeleton. Microfilaments are tiny rods of actin. They have the smallest diameter of the cytoskeletal filaments. They provide cellular movement, like muscle contraction. The microtubules, these are larger tubes of tubulin. They have the largest diameter of the cytoskeletal filaments. The rigidity maintains a cell shape. They make up the cilia and the flagella and centrioles, and they help move organelles. The intermediate filaments are composed of several proteins, cytoskeletal structures with diameter between those of the microfilaments and microtubules, and they support the nuclear envelope. Here we have an electron micrograph in color of microfilaments and microtubules. Okay, here we have a three-dimensional picture of these constituents. See the microfilaments here. Okay, other cytoplasmic structures, the centrosome. The centrosome means central body, and it consists of two centrioles. In the cytoplasm, it's near the nucleus. The centrioles are cylindrical, and they're composed of microtubules. The centrioles produce spindle fibers during cell division, which distribute chromosomes to the forming daughter cells, which we will talk about here in a little bit. Okay, here we have the electron micrograph of the centrioles. And notice if you look at the centriole from kind of longitudinally, you'll see it's composed of sections of 3 tubules each. Okay, cilia and flagella. Cilia are modal extensions of the cell membrane. It consists of microtubules and cylindrical patterns. It forms a fringe on the surface of certain epithelial cells. It's shorter than flagella, but it is very abundant when it's present. It beats back and forth in coordinated manner, and it propels mucus in the respiratory tract and propels the egg towards the uterus. The flagella, they are another type of modal extension from the cell membrane. They're similar in structure to cilia, but they're much longer. Flagellum causes the entire cell to move. We'll find these on the tail or as the tail of a sperm cell. It's the only flagellum we actually see in a human cell. And each cell has only one flagellum. Okay, this on the left shows a picture of cilia that you might see on the ends of certain cells, like in the respiratory tract. And on the right here, you see the tails, the flagella of sperm. Microvilli, these are tiny extensions of cell membranes, and they're composed of actin. They increase the surface area for absorption, like we would have in the small intestines. Okay, inclusions, they're present only in some types of cells. The chemicals are not necessary for survival. Some examples, melanin granules in the cells of epidermis or fat in the fat cells. And this is an image of the microvilli. Okay, clinical application, diseases at the organelle level. Malus, a mutant gene in the DNA of the mitochondria causes this, and these individuals cannot extract maximum energy from their nutrients. Crab disease, this is caused by the inability to produce one lysosomal enzyme. The person cannot produce myelin for the nerve cells, which leads to severe damage to the nervous system. The myelin is a coating on the axons and nerves. Okay, ADL, adrenoleukodystrophy. This is caused by the lack of a protein in the membrane of peroxisomes. Fatty acid buildup destroys the myelin sheaths of the nerve cells. And nerve cells cannot transmit nerve impulses fast enough in this case. Okay, moving on, the cell nucleus. It contains genetic material and controls cell activities. The nuclear envelope is a double-layered membrane that surrounds the nucleus. It separates the nucleoplasm from the cytoplasm. It has nuclear pores that allow passage of certain substances between the cytoplasm and the nucleoplasm. In the middle of the nucleus, we'll often see a nucleolus. It's a dense body of RNA and protein. It's a site of ribosome production. And then chromatin. This consists of the cell's chromosomes, each containing DNA, which is wound around proteins. And it stores information for protein synthesis. Okay, this picture shows the cell nucleus. And on the right, you'll see the electron micrograph. Again, within the nucleus, you see a nucleolus. And then floating around the nucleolus, you'll often see the chromatin, which is the DNA wrapped around the proteins. Okay, how do things move in and out of a cell? This includes passive and active mechanisms. Physical passive mechanisms do not require cellular energy, or ATP. That's why we call it passive. Examples: diffusion, osmosis, facilitated diffusion, and filtration. And we'll talk about each of those. Diffusion is the movement of atoms, molecules, or ions from a region of higher concentration to the region of lower concentration. So we're really talking about the movement of almost anything. It occurs due to constant motion of atoms in the molecules and ions. And it occurs only with substances that the cell membrane is permeable to, like oxygen and carbon dioxide and other lipid-soluble substances. A sugar cube dissolving in water is an example of diffusion. Solutes in water can diffuse across a membrane that's permeable to both of them until they reach equilibrium, which means the same on both sides. This graphic shows the sugar cube dissolving, and given enough time, those sugar molecules will disperse evenly. Okay, here we have diffusion through a membrane. As long as the membrane is permeable to that solute, they will move. A lot of membranes are not soluble to it, keep in mind. Okay, so if we have a membrane set up here in between two different solutions of a beaker, and on the left side, or A, we have a lot of the red solute molecules, and we have more of the blue water molecules on the right side. Over time, the red molecules and the blue molecules will even out on both sides. Again, that is diffusion. In this case, it is through a membrane, because it's simply high to low concentration movement of the molecule. Some examples: diffusion of oxygen and carbon dioxide between the blood and the cells, or between the blood and the cells of, say, the lungs. Okay, but generally, as long as there is more carbon dioxide in one cell and less in the bloodstream, the CO2 would move into the bloodstream, and vice versa. If there's more oxygen in the blood, it'll move out to a cell. So this is like the blood coming from the lung. Since it has a lot of oxygen in it, that oxygen will disperse into your tissues, and any of the waste product CO2 will come out and go to the bloodstream, and then that will be shipped to your lungs to get rid of the CO2. Okay, that's simple diffusion. Now, facilitated diffusion. This is still diffusion. It takes no energy, but it has to go through, say, a membrane protein like ion channels or carriers or transporters. It's used for substances that cannot diffuse across the selectively permeable membrane since they're not lipid soluble. Water soluble substances like sodium, potassium, chloride, glucose, amino acids, they can move this way. A passive transport mechanism is what facilitated diffusion is, with no ATP required. Okay, this graphic shows the movement of molecules through these carriers. Again, it is still basic diffusion, but we call it facilitated because it is helped. Next, osmosis. Osmosis is the movement of water. Keep that in mind. We're just talking about the movement of water across a selectively permeable membrane from a region of higher water concentration to a region of lower water concentration. Okay, don't get too confused here. That just means movement from where there's more water to less. But water is actually moving towards the higher solute concentration. So we often call this diffusion of water. Water moves into a region that contains higher impermeant solute concentration. And water moves through aquaporins. These are water channels between the phospholipids of the cell membrane. So water does not just easily move through a cell membrane. It has to move through these pores. Again, this is a passive process. No ATP is required. Okay, so here's an example of a membrane that is not permeable to water. Now, a while ago, what we showed was a membrane that was permeable to everything. Not true here. So, if we have a lot more water on one side than another, or we have a lot more solute on one side than another, and in this case, side A has a lot more solute, if you'll count the pink or red spheres. Okay, because this is a higher solute concentration on the left side, that means water is going to want to be pulled towards it. And indeed, that is what happens. It actually creates a pressure as it pushes or pulls, however you want to put it, the water through the membrane. Again, what wants to happen is for these protein molecules to move across the membrane. So generally, if this were diffusion and this membrane were permeable to the protein molecules would move towards B, but they can't move. So the only option is for water to move to the left, towards A. Now, as I said, a pressure can actually develop as a result of this. That's osmotic pressure. That's the ability of osmosis to generate enough pressure to lift a volume of water. Osmotic pressure increases as the concentration of impermeant solutes increases in a solution. And water moves towards solutions that have the higher osmotic pressure. Now, dependent on the solution that has, well, whether we're referring to the inside or the outside of, say, a cell, we can talk about tonicity. Okay, the ability of a solution outside the cell to alter the water volume inside the cell. So generally, we're going to refer to tonicity, or we're going to refer to the solution outside of a cell with tonicity. So an isotonic solution means you have the same osmotic pressure. That means you have the same concentration on the inside as outside of that cell. That means there's going to be no net movement of water. It's going to stay the same. A hypertonic solution, higher osmotic pressure. That's like a high salt concentration around a cell. So if you place a cell in a hypertonic solution, the cell will lose water. And it's like putting salt on the top of a slug. That will actually cause the slug to lose its water. It will shrink if you do that. Okay, hypotonic solution. That's a lower osmotic pressure. That means it's more closer to pure water. Cells in hypotonic solution will gain water. So if you put something in pure water, it can actually swell up, and it can actually even burst if we're talking about a cell that just has a membrane. Now, if you do this kind of test with a potato, it's really neat. The potato can be much harder if you soak it in water. If you put that same potato in salt water, the potato will get really soft because it will lose its water. Here we see the effects of different tonicities on a red blood cell. So if you put red blood cell in an isotonic solution, water just moves in and out equally, so nothing happens. And by the way, the inside of blood cells is usually 0.9% sodium chloride, so 0.9%. So in B, we actually show what would happen if we put that blood cell into a solution that's higher than 0.9%, like 5% or 10%. the blood cell would actually shrink and lose its water. Okay, this one shows the opposite. If we put a blood cell in pure water, the blood cell will take up water and it will actually burst. Okay, and then another type of movement of molecules or water with no energy is filtration. The process that forces molecules through membranes by exerting pressure. It's used to separate solids from water or small particles from large ones. An example, when blood plasma leaves the capillaries, water and small solutes will be filtered, but large plasma proteins in blood cells are not. Okay, they cannot move out. It's a passive process. No ATP is required. Okay, so this shows a capillary. And notice that some things can get out through the pore spaces, smaller molecules. Larger molecules, though, cannot move out, so they are filtered. Okay, so those were passive processes. Now let's talk about active mechanisms. They require cellular energy in the form of ATP to move the substances across the cell membrane. And we're talking about active transport, endocytosis, exocytosis, or transcytosis. So active transport. It's a movement of substances across a membrane from a region of lower concentration to a region of higher concentration. So this is the opposite of diffusion. It's going against a concentration gradient. It uses carrier molecules in the cell membrane, which are often called pumps. It's an active process. It does require ATP. Some examples: moving sugars, amino acids, calcium, hydrogen ions, sodium, and potassium with the sodium-potassium pump. There's also something called secondary active transport, where a carrier protein uses a sodium gradient to transport another substance across the cell membrane. Now, in this case, it does not require ATP energy. Okay, in this example, we have the sodium-potassium pump. It pushes out so much sodium as it brings in so much potassium. Okay, and later on in this class, we'll find out why the sodium-potassium pump is so important in establishing certain gradients, such as in the nervous system. Okay, this shows secondary active transport, where another molecule is moved at the same time as one other one. Okay, another form of, or an active process, endocytosis, movement of a substance into the cell inside a vesicle, like a bubble. Substances too large to enter by other methods can enter the cell this way. And there's three types of endocytosis. Pinocytosis or pinocytosis is when the membrane engulfs droplets of liquid. Phagocytosis is when the membrane engulfs solid particles. And then receptor-mediated endocytosis is when the membrane engulfs specific substances which have bound to receptor proteins on the membrane. Okay, in this first image, you can see a liquid droplet being brought into a cell. Okay, that's the pinocytosis. Okay, in the second example here, we have a vesicle taken in a particle in phagocytosis, and then it actually basically pushes it out of the membrane in exocytosis. Here we have receptor-mediated endocytosis. And like I said, the difference is it actually binds to some molecules before it's brought in into a vesicle. Okay, exocytosis, the release of substances or particles from cells. Vesicles containing particles fuse with cell membranes, and then they release the contents. Some examples are the release of neurotransmitters from nerve cells so that we can transfer a message from one neuron to another. We'll talk more about that when we talk about the nervous system. In transcytosis, This involves receptor-mediated endocytosis followed by exocytosis. It quickly transports substances from one end of a cell to the other. It moves substances across the barriers formed by tightly connected cells. An example would be the transport of HIV virus across the lining of the anus or vagina. And this image shows exocytosis, where we have a vesicle that's taken in a material and it lets that material out through the cell membrane. Here we have transcytosis. You see the material is brought in on one side of the membrane, then expelled on the other side. Okay, now let's switch gears a little bit and talk about the cell cycle. So when cells reproduce, lots of detailed steps are involved. And the purpose of this is to create more cells. So as you lose cells or as you're growing, we have to go through the cell cycle. And there are stages that include the interphase. That's when the cell is basically just growing, and it's the maintenance of normal function. So most of your cells are in the interphase. But when they decide they have to reproduce, they go into mitosis. Now, mitosis specifically means division of the nucleus. And then after the nucleus divides, the entire cell will divide. So when the cytoplasm divides, the plasma membrane, we call that cytokinesis. Okay, so this is the whole cell cycle. And as you can see, interphase comprises most of the cell cycle. Okay, again, this is when the cell is just being active and it may be growing some. It can be divided into G1, S, and G2. Okay, S is when new genetic material is being produced. This is when replication of DNA is occurring. Now, when a cell decides it needs to duplicate itself, then it'll go through mitosis, which has 4 phases: prophase, metaphase, anaphase, telophase. So interphase, this is an active period in the cell cycle. The cell's growing, it's maintaining the normal function. The cell replicates its genetic material right before it goes into mitosis. The cell also synthesizes new organelles and membranes and biochemicals to prepare for cytokinesis, which is division of the cytoplasm. But again, all of this is happening in the interphase. Okay, the phases of interphase, as I said, include S, G1, G2. S stands for synthesis. That's when the DNA is replicated. And that's going to happen before it goes into a mitosis stage. G1 and G2 are growth or gap phases. During these 2 phases, the structures and other molecules are being duplicated. Somatic cell division produces 2 daughter cells from an original cell. So your somatic cells are basically all your cells except for sex cells. Okay, mitosis, that's the division of the nucleus via karyokinesis. And then cytokinesis is the division of the cytoplasm. So the phases of mitosis include prophase, metaphase, anaphase, telophase. I'm going to describe them and then I'll show you pictures of them. So prophase, the chromatin condenses to form chromosomes. Centrioles move to the opposite sides of the cytoplasm. The nuclear envelope and nucleolus disperse. Metaphase, the spindle fibers from the centrioles attach to the chromosomes and align them midway between the centrioles. In anaphase, chromosomes are separate and they move in opposite directions towards the centrioles as the spindle fibers shorten. And telophase, chromosomes return to chromatin structure, the nuclear envelope forms around each chromosome set, and nucleoli become visible again. Okay, and this is a good table to get to know. Okay, let's look at the pictures, help understand a little bit more. So here is a cell basically in interphase. And we know it's an interphase because you still see of the nuclear envelope around the nucleus. So you still see the whole nucleus. Okay, prophase. Now we know we're entering prophase when the cell membranes, or I'm sorry, the nuclear membrane starts going away. And now you can see the chromosomes. So we say the chromosomes condense. And you start to see the centrioles. And the centrioles start forming spindle fibers. Okay, that's prophase. So it's getting ready. Oops. Okay, and here's late prophase. So you don't see the nuclear envelope anymore, and the chromosomes are kind of spread out, and the centrioles are totally on opposite sides. The metaphase. I always think of metaphase with the M in front standing for middle, because the chromosomes line up in the middle, and the centrioles, spindles have connected to those chromosomes. Okay, anaphase. I think of anaphase with the A as apart. And as you can see, the chromosomes are pulled apart. Now, each half of the chromosome, when they're together, is called a chromatid. So it's basically pulling chromatids apart. And let me say, each chromatid is exactly like the other chromatid. So essentially, each chromatid can be considered a chromosome when it's by itself. Now, telophase, we start seeing a new nuclear envelope forming around each set of chromatids. And finally, we go through cytokinesis, where the entire cell membrane divides. And that's a cleavage furrow when we're talking about an animal cell. And it divides into two brand new cells. Okay, cytokinesis. That is cytoplasmic division. Again, it begins during anaphase, and it continues through telophase. Now, in order for cytokinesis to work, a constriction forms, which we call a cleavage furrow. So newly formed cells will have identical DNA, but they may have slightly different sizes and numbers of organelles. Okay, so this shows each of these different levels of cytokinesis through an electron micrograph. Okay, so cell division needs to be regulated. We don't want cells all the time replicating themselves. As a matter of fact, that could be a big problem. That's what causes a tumor. Okay, frequency of cell division is regulated and varies by the cell type. So skin cells, intestinal cells, and blood-forming cells divide often and continually. Neurons divide a specific number of times, and then they cease. Okay, so neurons are some of the cells that aren't as easily reproduced. Kind of like once you have a certain number of neurons, that may be kind of it for a while, depending on where we're talking about. And the way cells know that they cannot reproduce anymore is that they have telomeres, or tips of the chromosomes, which keep shortening every time you replicate a cell. So there's a mitotic clock, and they can only replicate so many times. Fluctuating levels of certain proteins in the cell control cell cycle occur, and cells divide to provide a more favorable surface area to volume relationship. Hormones and growth factors are the external controls for cell division. There's also contact inhibition. Healthy cells stop dividing when they become crowded. So tumors can result from a loss of control over the frequency of mitosis. Okay, there's two types of tumors. There's benign and malignant. Benign tumors remain in the local area, and they often enlarge. Malignant are invasive, or what we call cancers, and they can spread or metastasize. Mutations in two major types of genes cause cancer. Oncogenes, abnormal forms of genes that control cell cycle, but they're overexpressed. Tumor suppressor genes, they normally limit mitosis, but if they're inactivated or removed, they cannot regulate mitosis. Okay, this shows electron micrograph of normal cells, and here we have cancer cells. Okay, and here we have characteristics of cancer cells. Now, the characteristics of cancer cells means that they do have loss of cell cycle control. It is heritable. It's transplantable. It has dedifferentiation, where you have loss of specialized characteristics. The cell just becomes some general cell that keeps reproducing. It has loss of contact inhibition. It also has the ability to induce local blood vessel extension called angiogenesis, where it basically creates its own blood cell or its own blood vessels. A lot of people have talked about cancers almost like an alien in your body. They are invasive, and they have the ability to metastasize or spread. Okay, stem and progenitor cells. Differentiation is the process of specialization of cells. Stem cells can divide to form two new stem cells called self-renewal. They can also divide to form a stem cell and a progenitor cell. And they can differentiate as any of many cell types. Progenitor cells are partially specialized stem cells. They're the daughter of stem cells. And they can divide to become any of a restricted number of cells that we call committed cells. Okay, totipotent. These are daughter cells that can specialize to become any cell type, such as a fertilized egg or cells of early embryos. Pluripotent are daughter cells that can become a limited number of cell types, such as stem cells of later development or progenitor cells. Here this shows differentiation of cells, going through those different levels of stem cells and progenitor cells. Okay, again, a stem cell can become almost any cell. The progenitor cell is limited. A lot of research being done on stem cells because we believe that we can make use of stem cells to create other cells that we really need to implant. Stem cells can be used to study and treat diseases. So stem and progenitor cells are required for growth and healing. In regenerative medicine, this is a field of study that uses the body's ability to generate new cells to treat diseases and injuries. It includes stem cell technology. Sources of stem cells for research, treating disease and injuries include donor stem cells like umbilical cord cells and stem cells from patients, either from natural sites like bone marrow or reprogrammed differentiated cells that are grown in cell culture. Okay, and finally, cell death. This is called apoptosis. This is programmed cell death. It's a normal part of development. It's a continuous stepwise process. For example, apoptosis happens when it removes webbing that's between the fetal fingers and toes. And yes, at one point of the fetus, you have webbing between your fingers and toes. This is a protective mechanism. It peels away damaged skin cells after sunburn, for example. Necrosis is cell death from damage. Now, that's not really considered as much of a normal process. Okay, this shows an outline of cell death, such as the individual had gotten sunburned and the skin peels off. And that is the end of the chapter on cells.